All right. Well, thank you very much for having me here to share some thoughts with you. I'm not sure if this is the title I had given you in the beginning, but I sort of rearranged everything and decided to do this talk in a completely different way than what I normally do. So let's see what happens. If anybody's interested in following up afterwards, everything is at this website, papers, software, everything is here. So let's start by just thinking about what this is. So here's an aqueous maze. Here's some kind of autonomous thing navigating this maze. It takes a corner without having to bump into the opposite wall. At this point, it makes a spontaneous decision to turn around and come back. So we might wonder what this is. And you've probably seen these before. This is a Xenobot, and here it is. It's been fixed down, and so the ciliary motion that it usually uses to swim through the water is, you can see it here, pushing the particles past it. And so one thing you might think from looking at it is, if you didn't know what it was, you might think that it's something we found in a pond somewhere, and you might think that it would have an invertebrate genome and so on. But it's actually a Xenobot. It's a biological sort of proto-organism and biorobotics platform that we made out of skin cells in the frog. But I think even more interestingly, it's a kind of vehicle for exploring anatomical morphospace. I see it as a tool, as a kind of telescope or periscope that allows you to peer into another space that we normally don't see at all. And it's a kind of visualization tool for diverse intelligence. And what I'm going to argue even more in this talk is that it's actually just one part of a highly heterogeneous composite entity, which consists of these biological cells, but also an AI, and also at least 4 human scientists. And so I think that's the right way to think about a Xenobot. Actually, is not just this thing, but actually as this composite entity. So what I want to do first is show you a little bit of biology and talk about some concepts, including multi-scale competency architecture, the idea that functionality in biology is often hidden. It's often cryptic because we get distracted by the reliable default outcomes. And I make this argument that in biology, every part is basically hacking all of the other parts to shape their behavior. And that gives rise to massive plasticity, virtualization, and the utility of a kind of software hardware lens. One of the basic things in our lab is that we add to the kind of easy way we often think of identifying intelligence in a familiar 3-dimensional space. And so we call this behavior, and we have these medium scale objects moving at medium speeds, and we're very good at recognizing those things as intelligent, apes and birds and octopuses and so on. But in biology, there are all these other spaces. There's a space of gene expression, and the space of physiological states, and anatomical morphospace, which is a space of possible physical configurations. Living beings navigate all of these spaces, and we don't have direct perception into this space the way we do in 3-dimensional space, and we're not very good at recognizing intelligence there. But it is ubiquitous in these other spaces, and I can only mention today a few examples. But one of the things that happens with being a navigating agent in one of these spaces is that you can get hacked. That is exploited and trained and communicated with. That changes your behavior. Now we're starting to be familiar with these kind of hacking examples in behavioral space. So, for example, zombie ant fungus that invades the body of these ants and gets them to do certain behaviors. It goes sort of higher than that. There's this parasite which has a life cycle in rodents that needs to get into a cat. And so what it does is it lowers the risk aversion in these animals, and they end up running around even though they smell cats, and then the cat will eat them. And humans that are infected with this tend to be things like captains of industry, and it really makes it actually makes people, as a whole literature on this, it makes people more willing to take risks, so more entrepreneurial. Lots of people who crash their motorcycle driving too fast for conditions and end up testing positive for this. So this is a very subtle effect on the behavior of a very complex system. And it's not that this parasite knows how to be an entrepreneur, it's that it knows how to hack the host to have these really subtle kinds of effects. And so now we can start to think about, what does that look like in these other spaces? So here's an interesting example. This is not Photoshop. I thought it was Photoshop the first time I saw this. This is a fly, which is running a kind of stripped down 2-dimensional ant program on its wings. And what it does, and the reason is that it sort of flaps these wings and it looks like the ants are scurrying around, and it keeps predators away because the predators don't want to deal with these stinging ants. And so this is kind of incredible. And then you think about, well, that's amazing, but even more amazing is the main program here that actually creates the whole fly. So this is quite something, but this is kind of a virtual kind of low resolution kind of example of morphogenesis. The main show is actually here. All of these cells are able to make different kinds of shapes. And one of my favorite examples is this. So you've got an acorn, and you ask yourself, well, what does an oak genome know how to do? Well, it knows how to make this. These cells can make nice flat green things. They do it very reliably. Every leaf looks basically the same, and so there you go. But it turns out that actually these exact cells can also make this, and they can make this. These are galls. They're induced by a parasite, a particular kind of wasp, and there are many other parasites, fungi, and so on, that can do this. And what it does is it reveals the fact that these cells with this exact same genome can actually leave the region of morphospace where all they do is make a flat green thing, and they can travel to this area. They're actually capable of making this. You would never know, right? By looking at how many oak trees can you look at without knowing that actually they're capable of doing this. So this is an example of biology hacking each other and revealing the latent morphospace and the latent physiological space of these cells, right? And so we really have to start thinking about this kind of software hardware distinction. Now, we look at something like this. So here's this frog, Xenopus laevis, that we use all the time, and it's got some kind of body, and it's got some kind of cognitive capacity and behavioral repertoire and so on. And we say, okay, well, here's an organism. And I want to make the argument that we only think this is an organism because we have these really compressed, pretty tiny lenses through which we see the world. We are very short-lived, very limited observers, and we see this animal, and we think this is the organism. But fortunately, we have synthetic bioengineering and things like that, and robotics, of course. And the Xenobot is one of them. It collapses a much bigger picture onto a scale that we can actually appreciate. In the case of the frog, this is actually the organism. It's this whole thing. It's the evolutionary cycle wrapped around all of the different things in the environment that end up producing the frog. So internally, of course, the genome sets some of the hardware, but also we've got various different degrees of agency in the environment that actually manipulate what ends up happening, from very simple kinds of chemical physical features, simple organisms, and very complex organisms like we bioengineers, right? So there's parasites, conspecifics, and so why is this the organism? Because you need to know all of this to optimize your ability to predict, control, understand, repair, and create, and in the case of more complex beings, to relate to it actually in both a functionally useful and an ethical sense. You need to understand what all the inputs and all the capabilities are. And so as we think about the Xenobot, so what is all this for the Xenobot? What does the broader conceived Xenobot look like? So let's just think about what Xenobots actually are. So pause this. So this is work that was done in collaboration with the University of Vermont. Josh Bongard is our partner for all this work. His student Sam Kriegman did the software, and Doug Blackiston, who's a scientist in my group, did all the biology for this. So what we decided to see was to really test the plasticity of cells and see what else they can do when liberated from their normal constraints. And so we have this frog embryo. We take these cells up here that are they're going to be skin. They're already determined to be epithelium, and we dissociate them and we put them in a little pile here. And now what could they do? Well, they could do many things. They could die, they could spread out, they could form a two-dimensional monolayer, they could do nothing. Instead, what they do is overnight they get together and they coalesce and they form this kind of thing. The flashes you see is calcium signaling, which is one of the things people track in brains, but there are no neurons here. This is just skin. And as a result, you get this. You get a system that is autonomously motile. So it uses these little cilia, these little hairs that normally spread the mucus down the body of the frog. And they have different kinds of motions. They can go in circles, they can patrol back and forth. They have collective behaviors, they have individual behaviors, they have lots of different kinds of behaviors. You can sculpt them in various ways. You can make ones that swim like this, like a fish. Here's Doug's magical donut swimming around. And if you track the calcium, you can do many interesting things. And this is still, the cognitive capacities are still being investigated. What do they sense? What are their preferences? What can they learn? What kind of learning capacity do they have? You can do all sorts of cool information theory on these calcium fluxes, exactly how you would do among regions in the brain, or in fact between two brains on a given task. You can ask what degree of coordination there is. They regenerate. So if you cut them almost in half, they will fold back up. Look at this hinge, 180 degrees. Think about doing like a bicep curl from a straight arm. Like look at the force that's required to fold this thing back up into its new Xenobot shape. And perhaps the most amazing thing so far, and I really think this is just the tip of the iceberg, the most amazing thing that we've seen is Josh and Sam discovered that in a simulation, in an in silico simulation, they discovered that if you sprinkle a bunch of loose material in the vicinity, what they would do is through their actions, they would displace this material in a very interesting particular way. And so part of the deformations and the stimuli that we give these bots come from this virtual world, this AI. It's an evolutionary wrapper around different shapes and different ways to make these bots, and it can make predictions. And Doug, amazingly, Doug can actually make some of these. And so what we found is that if you do provide them with particles that are themselves an agential material, so not just loose, kind of a passive matter, but actually an agential material, what they will do is they run around and they coalesce it into little piles. The little piles, and they make them sort of tighter and tighter. The little piles, because they're an agential material, this is what enables the next phase, which is that it itself becomes the next generation of Xenobots. They mature, and guess what they do? They start to run around and do exactly the same thing. And so that gives you the second generation, the third, and so on. So the reason this works is for the same reason that the Xenobot construction works in the first place, because we worked with an agential material, which are these cells that have an agenda of their own, and they're able to do the same thing. And so this is kinematic self-replication. This is sort of von Neumann's dream. This is some sort of robot that can go around and make copies of itself from materials it finds in the environment. As far as we know, no other creature does this. So we'll get back to some of the implications later, but this to me is a Xenobot. It's not just this. It's not just the actual thing here. The whole cycle that allows these Xenobots to exist involves all of these components. It involves these other intelligences, meaning us in the environment. It involves the cells. It involves the genome of the frog that made some of this hardware. It involves part of the morphospace that is this novel aspect here. It involves the AI actually that was helping us to determine the optimal stimuli and optimal shape. And so this, I think, is a composite entity, and I think we need to really get behind this idea that beings are not just simple physical things that are obvious to human observers. This whole thing is the Xenobot. Okay. And that's kind of strange. We're not used to thinking these highly heterogeneous. I mean, some biologists that study weird symbiotes and so on, they're sort of on their way. But typically we don't think about these really strange composite kinds of entities as a single thing. But we should, because you and I are also collective intelligences, not just ants and bees and bird flocks.